Genetics students often study a very important set of experiments which map bacterial chromosomes, taking advantage of prototrophic strains of bacteria, which could make all their own nutrient requirements, and auxotrophic mutant strains which needed to be supplied with a specific nutrient in order to survive. If a genetic exchange called conjugation with an HFR strain possessing a functional gene to synthesize a nutrient occurred with an auxotrophic strain which lacked a functional gene, one could use this then to map a bacterial chromosome, as I will explain in other videos. How could you set up an experiment so that the only bacteria which could grow on, say, a plate which lacks histidine are those which have undergone conjugation and have received a gene for histidine metabolism? Well, certainly auxotrophic strains which lacked genes for histidine synthesis would not be able to grow. But if part of the procedure was to combine them with a strain which was the source of the HFR plasmid, would not these HFR strains be able to grow on plates without histidine, since they are the source of the histidine synthesis gene? Well, one could perhaps try to set up a scenario where they could not survive. Let's say that this HFR strain was an auxotrophic strain which could not synthesize something else, like biotin, while the recipient strain could. If all of the plates lacked biotin in their media, then the source of the HFR strain itself would not be able to survive on those plates. This would be a way of controlling for that. Here is a second problem. How could you prevent the gene for biotin synthesis from traveling from what we designate as the recipient strain to the donor strain? Well, at first this is easy simply because HFR plasmids are transferred in one direction, and so there would be no transfer from the HFR recipient strain to the donor strain at first. But if the recipient strain receives a whole HFR plasmid, then reverse transfer could be possible. So this would be a problem if one was not maintaining one's bacterial stocks well after repeating this experiment, and so you would need to screen your stocks from time to time to make sure that no changes had occurred. Here is another problem. If it was mutations which rendered bacteria not able to synthesize their own biotin or histidine or lysine or whatever, how does one account for or control for new mutations which would then revert these mutant bacteria that are auxotrophs back to the wild type condition where they would be prototrophs? Well, two scenarios might be: one, work with stocks which had deletion genes. So if a mutation caused a simple change at one nucleotide to make it non-functional, then a subsequent mutation could possibly change it back to the wild type condition, which would be functional. However, if the mutation caused the loss of the gene or part of the gene, then no single point mutation could return the wild type function. Or perhaps one could simply then calculate what is the frequency of reversion mutations back to the wild type condition, say 1 every 10 million, and thus one would, as you score a positive result in your experiment, simply then need to see a rate higher than 1 in 10 million. You would then be, say, accounting for that. These were just a couple of examples in how an experimenter must carefully design their experiment and choose specific strains of their research organisms, such as specific types of mutations, so that the results actually do show what they attempt to show.